today's video we're going to discuss about the conditions required for plant growth and the concept of differentiation. So first let's start with the conditions. Conditions required for plant growth. The first one is oxygen. So oxygen is very essential for plant growth because oxygen is very important for the aerobic respiration. So we know that what is aerobic respiration. Aerobic respiration is where it's a respiration where oxygen is present. So it's aerobic respiration, and this aerobic respiration produces ATP, which is important. This is the energy which produces or which provides energy to the plants to do different activities. So oxygen, aerobic respiration, and ATP. ATP is very important. The second important condition for plant growth is water. So for example, there is a cell, and when water goes into that cell, the cell becomes turgid, which means that it enlarges. So the cell becomes turgid. It is for turgidity, and when the cell becomes turgid, there is vacuole enlargement. Vacuole enlargement. So water is also very essential, and this vacuole enlargement also helps in cell growth. When vacuole enlarges, the whole cell also becomes elongated, that is cell growth. So first one is oxygen, second one is water. The third one is nutrients, that is micronutrients and macronutrients. These nutrients are very helpful for protoplasm, for synthesis of protoplasm. So we know that there are two kinds of nutrients, that is micronutrients and we also have macronutrients. These are the nutrients which are required for these plants. Nutrients are the minerals. So when we talk about nutrients, we're actually referring to the minerals which are present. So micronutrients and macronutrients, and these both are required for synthesis of protoplasm. So this is the third one, that is nutrients. The fourth one are the hormones. So hormones are also required, which are called as phytohormones. Phytohormones are the plant growth regulators. And here also we know that there are two kinds, that is inhibitors, or let's say first promoters and then inhibitors. So here we can write these phytohormones are the plant growth regulators. Here we talk about auxins, gibberellins, and cytokinins, ethylene, and abscisic acid, all those things. So that those are phytohormones, which are very important. The fifth one, which is important, is temperature. Optimum temperature. So this optimum temperature is very important for the optimum enzymatic activity. So for enzymatic action or enzymatic activity. So till now we have learned about oxygen, water, nutrients, phytohormones, temperature. Sixth one we talk about is light. So light is also very essential, and the main process in which plants use light is photosynthesis. So photosynthesis is where plants make glucose or food using the sunlight. So these are the six main conditions which are required for plant growth. We have oxygen, water, nutrients, that is minerals, phytohormones we have, and temperature and light. So these are the six conditions which are required for the plant growth. So this is about the conditions. Now let's talk about the concept of differentiation, de-differentiation and redifferentiation. So now let's talk about differentiation. Now for this we have to learn about cell cycle. So basically we know that what is cell cycle. So here we have the cell cycle, and we have like this and like this, somewhat like this. So here, so it's a bit bigger, so here like that. So this is the G1 phase. We know these G1 S, G2 and the G0 phase as well. So this is G1 phase, this is the S phase, this is the G2 phase, and this is the M phase. Now we know that G1 phase, S phase and G2 phase is collectively called as interphase. So this whole thing is known as interphase. So it is interphase, G1, S and G2, and we have M phase. Now what happens here, if a cell is there and it divides into two daughter cells, here sometimes when this cell cycle happens, the first cell comes from here and then it goes here, and then suddenly it comes out of the cell cycle, and enters the G0 phase or G0 phase and turns into parenchymatous cells. Parenchyma. It turns into a parenchymatous cell. Now, it turns into parenchyma cell. So here we'll draw a little diagram. We know that we learned about xylem and phloem and the medullary rays. So here we know that there are vascular bundles, and this is the fascicular vascular cambium. So here there is xylem, phloem, and we know that there are these elongated parenchymatous cells between these vascular bundles, which are called the medullary rays. So they are very elongated because of the lack of space. So they are like this. So we have to label it. This is known as the fascicular vascular cambium, and this is known as, these cells are known as medullary rays, which consists of parenchyma. So these are basically the parenchyma. So here what happens is they come out of the cell cycle and forms the parenchyma. So let's label it as 1. Again these parenchyma cells again enter the cell cycle. This let's label it as 2, and again when they come back to the cell cycle, again they come out of the cell cycle and enter the G0 phase, which we call as 3. So we are just labeling it for our understanding. So the first one is differentiation. So differentiation is basically when the particular cell which is undergoing cell cycle, it comes out of the cell cycle and enters the G0 phase and forms and turns into parenchyma cell. So that is called differentiation. It's the first stage. Then what happens? Again the parenchyma cells enters the cell cycle, and this is known as dedifferentiation. And then the third one which we have labeled is redifferentiation. So in simple words, what we can say, there is parenchyma cell, or let's say we have the permanent cells and the meristematic cells. Let's talk about like that. So we have meristematic cell and we have permanent cell. When this meristematic cell, so permanent cell, the simple permanent cells are parenchyma, collenchyma and sclerenchyma. These both are simple ground permanent tissues, and complex ones are xylem and phloem. Those we are not talking about here. So when this meristematic cell turns into a permanent cell or tissue, this process, when this meristematic tissue turns into permanent tissue, this is known as differentiation. When this permanent cell turns back into meristematic cell, it is known as dedifferentiation. When this meristematic cell again becomes a bit differentiated and forms permanent cell, it is known as redifferentiation. So there are three important terms that we need to remember: differentiation, dedifferentiation and redifferentiation. Differentiation is when meristematic cell becomes differentiated and turns into permanent cell. Differentiated means specialized for a particular function. That is known as differentiation. Again, this permanent cell, it again turns back to meristematic cell. It is known as dedifferentiation. Again, meristematic cell, it again turns into permanent cell. It is known as redifferentiation. Permanent tissue, permanent cell. So we can also understand it by the cell cycle. When it leaves the cell cycle, it is known as differentiation, and when it comes back again, which we label it as 2, that is dedifferentiation. Again the cell leaves the cell cycle and enters the G0 phase, that is 3, redifferentiation. So these are the three important terms: differentiation, dedifferentiation and redifferentiation. Now let's talk about one more very important term, that is plasticity. What is plasticity? Now let's take some examples. In case of cotton, coriander and larkspur, so let's write it here: cotton, coriander, larkspur. In these kind of plants, the leaves of these plants change during different times of the year. So they have different leaves according to different seasons and different parts of the year. This we know is called as heterophyll. So this is known as heterophyll. In this case, it is leaves which are different throughout the year. So these are leaves we are talking about. This is what is known as heterophyll. Now what is plasticity? Plasticity is basically the ability of the plants. So we can write here, it is the ability of plants, so it is the ability of plants to respond to external stimulus or stimuli. So when they respond to the external stimuli based on different seasons by showing different structures in their plant body. So by showing different structures. So here we told the structure is leaves in these cases, which is known as heterophyll. Cotton, coriander and larkspur, in their plant body, the leaves are different throughout the year. That is known as heterophyll. Here the leaves that are different. So basically plasticity is the ability of plants to respond to external stimuli by showing different structures at different periods of time. So at different parts of year, we can tell it like that. So different parts of year, not only year, throughout their life. So let's write it like that. So some plants, when they are young, they have different kinds of leaves, like these examples, and when they grow up, their leaves become a bit different. So by showing different features at different ages or different periods of time. So this is what plasticity is. It need not to be leaves. It can be whatever structure it is. It can be fruits, flowers, roots or stem or whatever it is. So basically plasticity is the ability of plants to respond to external stimulus by showing different structures in their plant body at different periods of time. So this is known as heterophyll. Heterophyll is different from plasticity. Heterophyll is where the leaves are different during their lifetime. So that is, examples are cotton, coriander and larkspur. So this is what plasticity and heterophyll is. So in this video we have discussed about the conditions for growth, which is water, which is oxygen, water, nutrients, that is minerals, macronutrients and micronutrients, the optimum temperature, which is for optimum enzymatic activity. Light is also required for photosynthesis, and also very important phytohormones, plant growth regulators. And we have also discussed about this one, which is differentiation, dedifferentiation and redifferentiation. So basically we can tell that, for example, if there is a cell and it divides into two cells, and then these cells become big, so a bit big, and basically they get specialized to perform a special function. They get differentiated or they become mature. So that's what we are talking about. So basically differentiation is when meristematic cell, or when a simple non-mature cell, it becomes matured and specialized for a special function, that is known as differentiation. And then when the permanent cell again turns into meristematic cell, it's known as dedifferentiation. Meristematic cell again turns into permanent cell, that is redifferentiation. This is a simple way we can summarize the whole information. So this is about the differentiation and redifferentiation concept, and we have taken a look at this one. So basically the parenchyma cells are the medullary rays, which are between two vascular bundles. And we have also discussed about the term plasticity, which is the ability of plants to respond to external stimuli by showing different structures in their plant body at different periods of time. So this is all about the conditions for growth and the differentiation concept.